Okay, welcome everybody. Today's lecture is going to be about tRNA and protein synthesis. You'll have to bear with me this morning. It's awful early, but I think we can rock this one out. So let's go ahead and get started. Let's start with tRNA. tRNA, what does tRNA stand for? Transfer RNA. So what's its function, do you guess? It's going to transfer an amino acid to a growing amino acid chain, also known as a peptide chain. And that peptide chain is going to be our protein. Proteins are made out of peptide units. We're going to build that protein with the building blocks of amino acids. And this tRNA, transfer RNA, is going to transfer those amino acids onto our growing peptide chain. And we'll cover that process. Things that I would like to point out, there are three different types of RNA. You've got tRNA, which we'll talk about. We've got mRNA, which we'll also talk about a little bit. And then also you've got rRNA. The stuff that I would like to highlight is your tRNA is going to be your smallest, generally made up of less than 100 nucleotides long. So let's go ahead and get started. RNA, tRNA. See if I can find a better marker. Where do we begin? Well, here's going to be the structure of our tRNA molecule. It kind of looks like a clover shape. There's different ends to it. So we've got a 3' end and a 5' end. We all know that DNA has a 3 and 5' end. So this is going to be our 3 and our 5' end of our RNA. So off of this, we also know that at our 3' end, we're going to have a CCA triplet. The CCA triplet. And what that's going to do is it's going to accept an amino acid onto that. So this is going to be my amino acid just for drawing sake. We're going to attach an amino acid to the 3' end, which happens to be a CCA sequence. And then opposite of this 3' end, down here at the base of the 3-leaf clover, we've got an end. And this is going to be our anticodon end. Anticodon. So down here at the very tip, we've got a little sequence end. So if we kind of take that sequence that's right at that end, it could be a it would be a triplet. So it would be like a CCU, GCA, whatever it wants to be. However, we'll find out later that it is specific, but just notice that it is a triplet. There's three different codes here. So what else can I say about this? We do know that it is carrying the amino acid. We know that its structure is a clover. Then we also know that it's going to be recycled within the body. And that's going to be an important concept because every time we want to create a tRNA, we're going to recycle it and instead of having to create a new one every time. So now let's kind of get a little deeper into the science. So let's draw a naked tRNA. That end's a little shorter. We've got our 3' end, our 5' end. Let's just draw another one really quick. Same thing, 5', 3' end. Oops, prime end. How do we go from a naked tRNA molecule to one with a amino acid attached? Well, we're going to need an enzyme. And the enzyme that's going to be required is going to be called aminoacyl tRNA synthetase. So let me write this in. Aminoacyl tRNA synthetase. All right. I circle it like that because that's how I do all my enzymes. And this enzyme is going to catalyze the reaction of breaking down an ATP, an adenosine triphosphate, meaning it has an adenosine with 3 phosphates attached to it. So 3 phosphates, that's shorthand for my phosphate. And it's going to spit out an AMP, so an adenosine monophosphate, then also a free phosphate as well. So what does this mean? We're going from a triphosphate to a monophosphate. Well, within each of these phosphate bonds, we hold energy. We're going to cleave off 2 of those bonds, and we're going to use 2 bonds worth of energy to stick a free amino acid within the cell. There's a free one. We're going to take this energy. We're going to attach our amino acid onto the 3' end, and that's going to be specifically the CCA sequence. That's going to be here. So now we're left with an active tRNA molecule. So this is going to be active. One thing I would also like to point out is this aminoacyl tRNA synthetase enzyme is going to make sure that the correct amino acid is attached. So how does it do that? Well, we're going to have to look down here at the sequence now. Remember, this is our sequence of interest. It's going to be our anticodon region. And I will explain a little bit about what anticodon means in a little bit. But what this enzyme is, it'll make sure that the correct sequence down here matches the correct amino acid that's attached to it. So, let me just make sure the camera can see what I'm talking about now. That should be good. So we have our active, get a marker that works, that'll do. So we have our active tRNA, and what are we going to do with it? Well, we are going to put it onto its respective mRNA sequence. So this is going to be mRNA. And like over here, we have a 3' end and a 5' end to all RNA. So let's assign a 5' end and a 3' end, and this will be just basics. I want you to understand what we're doing right now. We're going to be taking a tRNA, sticking it over here, and then we'll be adding this amino acid that got added to our active tRNA, and then we'll make it into a peptide chain. So keep that in mind. But for right now, our active tRNA molecule is coming along, recognizes the sequence that it wants, also known as a codon, which is going to be a triplet. So it's going to be like a CAU, a GCA, whatever combination you want. There are 64 different combinations of genetic code. For the example, off the top of my head, this should be a histidine. A CAU sequence should be a histidine. So what are we going to see here? Well, we have our 3' end, 5' end. Notice how the 3' and the 5' go together, and the 5' and the 3' go together. You'll see that a lot in DNA and genetics. 3' and 5' always go anti-parallel to each other. Okay, but so we've got our activated, because we've already activated our tRNA through aminoacyl our tRNA synthetase. So we've got this tRNA and this codon. Well, let me get a different color here. Remember I said this sequence is the anticodon. It's going to be exactly opposite of our CAU sequence. So what is it going to spit out? It's going to spit out a GUA. So this sequence down here is going to be a GUA. So like I said earlier, that enzyme that was used to make this active molecule is going to make sure that a histidine is attached. Because it's going to look down here, it's going to see a GUA, and it'll think opposite. So it'll think a CAU. So it'll attach a histidine, specifically a histidine amino acid. So that way when our active tRNA finds its anticodon sequence, when it matches up down here to our codon, it's going to add this amino acid to a chain that's already formed. And we're just going to form a peptide bond, a covalent bond, and then add this amino acid to our chain down here. It gets rid of its amino acid now. Now we have a naked tRNA, and then it's going to leave. And then our next one is going to come, find its sequence that it's going to bind to, the anticodon-codon sequence. It's going to attach its amino acid and continue. So that's the very, very basics of this sequence. So let's get into a little more expanded view now. So actually I can leave that up. Make sure my camera's catching everything here. All right, so now we're going to move on to an expanded view. So again, we've got our mRNA, we've got a 5' end, got a 3' end. I warned you it was a little early in the morning. So we've got our 5' and a 3' end. Now let's get a little more expanded here. So we've got different sequences. We've got an initiation phase, we've got an elongation phase, and a termination phase in protein synthesis. Because remember, our ultimate goal is to make a protein. So that's our final goal. So what we're going to do is we're going to attach our ribosome. And a ribosome has ribozyme activity, so it has an enzymatic activity to it. So there's two different subunits. There's going to be a 40S and a 60S, which make up the 80S ribosome, and that's going to be found in eukaryotes. Prokaryotes, on the other hand, are going to have a 30S, 50S, 70, which make up their 70S ribosome. So think evens, eukaryotes, odds, prokaryotes. So that's important to remember, especially when we get to talking about drugs in just a little bit. Right now, we're just focused on the human for now. But remember, 40S, since 40 is less than 60, this is going to be the small subunit. Since 60S is larger, it's going to be the large subunit. And then the 80S complex is going to be both of those together. So, somebody didn't do their math, but this is just traditionally how it's done. So first off, our 40S subunit binds. This is going to be the initiation phase. Initiation factors and GTP. So initiation factors and GTP are both required for this 40S subunit to bind to our mRNA sequence. The mRNA sequence is going to be our messenger RNA, and it's going to be the longest. They're going to be long, long codes. So we've got our initiation phase. So next what's going to happen is we're going to have our large subunit come in. That's going to be our 60S, and it's got three different sites. It's got an A site, give it a different color. It's got an A site, it's got a P site, and it's got E site to it. So our 60S subunit has an APE kind of area to it. So what I would like to focus is just the basics of what's going on by incorporating our tRNA into our ribosome and then eventually our amino acid. So we're going to look at a little snapshot of a tRNA molecule coming in. It's not going to be the very first one to come in. A lot of other tRNAs have come in before it. They've already dumped off their amino acids into this chain. So let's draw this out. So we've already got a few amino acids onto our chain. This is our peptide chain, so it's a polypeptide chain at this point. We've already got there we go. So we've got a tRNA already here we're going to look at a little snapshot. Then we've got an empty tRNA. This one has already dumped off its amino acid to the chain. It moves over to the E site, the exit site, and this tRNA will eventually leave. And then it might go off and get another amino acid, wait along for its sequence to appear down here. When it finally appears and it's activated, it'll come back. So now we've already exited that. That one's gone. We've got an tRNA with an amino acid chain building upon it. So what we're going to do is we're going to pretend we just activated a tRNA, and it's going to come along, and now it's in the CAU sequence. We've got a CAU sequence. We've got our tRNA that we've just activated with its amino acid, and we've got a GUA sequence, the anticodon sequence, that'll match our codon sequence. The codon is going to be found on the mRNA. It'll be opposite of our anticodon sequence. So this tRNA is going to enter the A site. Think of it as an acceptor site. So it's going to accept the tRNA. So next, we're going to go to elongation. Elongation simply refers to we're elongating this chain here. We're adding amino acids to our polypeptide chain. So, sticking with colors, we've got our tRNA with our histidine molecule attached to it. It enters the acceptor site, the A site of this ribosome subunit. So, we have an activated tRNA ready. We've got our chain that's already been made. Now, let's use ribozyme activity. Remember I said that the ribosome has inherent ribozyme activity, enzyme activity. What it's going to do is it's going to catalyze the reaction of attaching this amino acid onto our chain. So to do that, we're going to form a peptide bond, a covalent bond. We're going to shift this tRNA over, and this tRNA is going to shift over again. So let's draw this out. Kind of a deformed looking one. We just shifted over. We just added our histidine molecule. That was our histidine. And now we're going to be attached to that one that we just activated and moved over. There we go. So we're expanding this polypeptide chain. So it just got one amino acid longer. So formed a covalent bond. Now let's talk about the stoppage, the termination. There we go. Termination of this chain. So let's say we finally get to a sequence that says, let's say a UAG. So a UAG is going to be a stop sequence, a stop codon. There are going to be three stop codons, the UAG, UAA, and UGA. Those are going to be your three stop codons. So once we have a stop codon here, we realize there's going to be no more amino acids attached here. The polypeptide chain is done. The subunits will leave. Subunit will leave. We have this beautiful polypeptide chain that is now going to be used within the body somewhere, hopefully, and we are done. So that is going to be the protein synthesis pathway. So that's kind of the basics. Release factors are required for this. That's a little more complicated, but just realize that you have three phases. You have that initiation phase, which is going to be this, which is going to be the 40S subunit binding to our mRNA. Eventually our 60S subunit will come in Then we've got our elongation phase. That's going to be where we are elongating this polypeptide chain. And then finally, we've got the termination phase, and that's going to be where our stop codon here, the UAG, UAA, UGA, and that's where we dissociate everything from the mRNA. It's done its job, and now we are left with a polypeptide chain. So that's kind of the basics. Now let's get into some of the antibiotics used. To do this, I'm just going to put it in the center and we can get rid of this basic structure here. Alright, so hopefully make sure we can see over here. Let's use black. Black. Okay, we've got some different antibiotics here. We've got tetracycline. Tetracycline. What is the action of tetracycline? Well, it's going to bind to the 30S subunit, so 30S, and make it so the tRNA cannot bind. So tetracyclines are going to bind to 30S. Well, look over here. We've got a 40S and a 60S, but remember, this is in eukaryotes. So in the human body, tetracycline isn't going to affect our ribosome subunit. Instead, bacteria are the 30S, 50S subunits. So tetracycline is going to bind to the 30S, the small, since 30 is small. 30S is going to be the small subunit, so we can't add tRNAs, which means our polypeptide chain can't grow, and that's effectively going to be how this antibiotic works, the tetracyclines. There are other antibiotics like aminoglycosides, which also bind that's not going to show up. Aminoglycosides. That's also going to bind the 30S subunit. However, mechanism of action is going to be a little different. It's going to block the initiation step. So we're going to block the 30S subunit from interacting with this mRNA, blocking the initiation step. Remember, we have the initiation step, the elongation step, and the termination step. Initiation, elongation, and termination. Aminoglycosides are going to block the initiation step. So we can't even begin to make our polypeptide chains. All right, next we've got chloramphenicol. Chloramphenicol is going to block the 50S subunit. Chloramphenicol is going to block the 50S subunit. So remember, now we're still talking about prokaryotes. Now we're going to affect the large subunit, and it's going to block this step right here. We're going to block the transfer of an amino acid. Man, I got to find a better marker. Going to block the transfer of an amino acid to our polypeptide chain from a covalent peptide bond. We're going to cancel that step. We can't form a covalent bond between our polypeptide chain and our amino acid because chloramphenicol is going to block this 50S subunit. Next, we've got the macrolides. Macrolides are going to block the elongation process of protein synthesis. So like I said, we've got 3 different phases. Now, macrolides are going to affect the elongation. So we're not able to elongate this chain as well. So it has kind of a similar effect to the chloramphenicol antibiotic. So we have macrolides. Remember that we're not affecting humans. We're only affecting the bacteria as well. Then finally, we've got our clindamycin, and that's also going to have a similar effect as chloramphenicol. So clindamycin. All right, so these are some of the basic antibiotics that we're going to be using and how its effects are affecting protein synthesis. So look, different mechanisms of action. We have a really complex process going on. Here's the eukaryote. Just as easily, we can make a few changes and turn it instantly into a prokaryote. There, prokaryote. That was pretty easy. Same process. Both humans, eukaryotes, and prokaryotes use the same ribosome process, just different subunits. There's also a Shine-Delgarno sequence in the 30S. That may be a different video that I'll make. However, we're just talking about the basics of an amino acid chain synthesis. So we've got our tRNA, our ribosome. The ribosome has ribozyme activity, meaning it has an enzymatic activity, which will catalyze the bond between these two. We have our tRNA, our growing chain catalyzes that. We have a whole bunch of antibiotics that can disrupt that process at different phases in the process. Macrolides block the elongation of this chain. Clindamycin and chloramphenicol both block the covalent bond formation. We've got aminoglycosides, which act upon the initiation phase, so we're not even able to start this process. And then finally, we've got our tetracyclines, which will affect the 30S subunit as well. So hopefully this makes a little sense. If you have any questions, be sure to ask. Otherwise, be sure to like, comment, and subscribe. Thank you very much.